This is component 1 mock, and it's mock number 3, and we're on to question 3. Here's a diagram showing stages in respiration. You should have learnt this diagram and know just about everything about it already. The question: ATP is produced by phosphorylation during both anaerobic and aerobic respiration. Name the types of phosphorylation at A and B. So we have got A, which is there, and then another A, so don't miss the other A. Then we've got various B's coming off. So A is at substrate level phosphorylation. So substrate level phosphorylation. That's because we're still breaking down our substrate. So there's our substrate, our sugar, and our sugar 6, 3, 2, and then 5, 6, 5, 4. So we're breaking down our substrate sugar there and generating ATP directly. So that's substrate level. Whereas the other ATP, on this diagram, it's actually shown in brackets. So it's going off to make ATP somewhere else, and that's in oxidative phosphorylation. So that will be oxidative there. And that's after the substrate has been broken down. Part B, state the type of enzyme. So not the actual name of the enzyme, the type of enzyme that removes either carbon dioxide or hydrogen. And you should be okay with these. These are D, meaning removal of carboxylase, so removal of CO2. And the second one again, D, hydrogenase. And you'll have done experiments with utilising those enzymes. There are 4 places where hydrogen acceptors remove hydrogen in the Krebs cycle. Name the hydrogen acceptors. Well, in respiration, you've got NAD and FAD. In photosynthesis, you've got NADP. So we just need to get them right. Well, the clue is there's lots of X's, so that's our most common one, NAD. And just once on the Krebs cycle, the odd one there. And another clue is it's 2 ATP produced rather than the 3. So another little clue that it's not NAD, so FAD. You can't say reduced here or NADH because it says name the acceptor, so presumably it hasn't accepted it yet, so you need to say NAD. Explain why these 2 hydrogen acceptors lead to the production of different numbers of ATP. So here we've got NAD makes or leads to 3 ATP being produced, FAD 2. And it's just due to the number of pumps in the kind of carrier system. So for example, FAD carrier system has 2 pumps, so 2 of those proton pumps. NAD has 3 pumps. And then complete the table. Now you don't need to know all of this because you can get it from the diagram. From 1 molecule of glucose. So that's something you need to bear in mind because some of this happens twice. So in our link reaction, what have we got? There's our link reaction. We are making 3 ATP via NAD there, and that happens twice. So that will be 6 in total, which is our 3 ATP times by 2, because this 6 carbon sugar has been split into 2 pyruvates, and then this bottom half happens twice. So that's why sometimes we call it the Krebs bicycle, because there's 2. Any ATP made directly there? No. And so our total will be 6 there going across. Krebs cycle, we've got a 3, a 3, a 3. Those are our NADs. So our NAD will be 3, 3, 3 will be 9. Then we've got our FAD, which will be a 2. And that will happen twice. So that will be 11 times by 2 will be 22. How many ATP molecules are formed directly? Well, looking through here, we just got the 1. But of course this cycle happens twice per glucose, so that's going to be 2. And then when we add that up, that's going to be 24. Identify molecule Z. Where's Z? Oh, there's Z. So, and you should be fine with this. It's the pyruvate gets converted to it, and then that enters our Krebs cycle. It must be a 2 carbon, because a 4 plus 2 is 6. And you chemists will know that acetate or acetyl, that's sort of the 2 carbon word in chemistry. And so the answer is acetyl coenzyme A. Acetyl coenzyme A, or you could say acetyl CoA as an abbreviation of that. State the number of carbon molecules. So even if you didn't know the name or hadn't heard of it before, you could guess it. The 3 minus 1 or 4 plus 2 is 6, so it's going to be 2. Describe the formation of molecule Z. So how is it made? So just kind of say what you see here, really. So we're going to say pyruvate is converted to a 2 carbon, which will be kind of there, and then it's going to have the hydrogen removed as well. And we're going to say the other thing that appears as well. So we're just going to try and remember our steps there. So pyruvate, that's our starting position, which is 3 carbon, converted to the 2 carbon acetate, converted to acetate, which is 2 carbon, or you could say an acetyl group possibly. What's happening? We are removing CO2. So by removal of CO2, or we could say decarboxylation, we could also talk about the hydrogen. And then the last little step is that acetyl group, or the acetate, is combined with something else called CoA to form our acetyl CoA. So acetyl group combines with coenzyme A, and that will then form molecule Z, which is our acetyl coenzyme A. So a little bit of a tricky finish there, but you should have, if you'd learned that, you should have got the vast majority of those 15 marks.